Yes.
So, Dr. Young is a professor of the Department of Medicine and Division of Experimental Medicine at McGill University in Montreal, Canada. And he recently performed a large study on KAT6A, KAT6B, and he'll tell us more about his study.
Hello, can you hear me?
Yes.
Okay. So it's challenging for me to speak here. I'm really interested and really happy to be here. So because the audience is quite different from the typical ones I speak to. So I will try to make the scientific slides more accessible to you. I was, for my university study, I was a chemist. I studied chemistry, so then I got a PhD degree in Shanghai and then went to NH, which is very close here for training. After that, I went to Canada for my faculty position. So my research has been mainly focused on how the genetic material is organized and regulated, especially about epigenetics, as mentioned by the previous speakers. For today's talk, I will be mainly focused on one protein and its related protein, KAT6A and B. And that one protein is called BRPF1. But before that, I will give you a little bit introduction. So one thing I found is that most of those mutations are de novo mutations. So this is new to me, because this is relatively new to me. So how does this occur? This is random. This basically is biology, nobody's fault. It's just like a flip a coin. You got a head or tail. It's a random process. So that's important. Basically nobody's fault. So that's very important. Because when I speak to some of the parents, they ask me, what's going on here? So this is, I think it's very, very important. I remember I went back to my home village in China. So there was this family and a distant relative with 2 kids. Both have muscular dystrophy. And they try to figure out what's going on, whether they did something wrong. And of course, in that case, I still don't know, because parents are normal, maybe due to recessive mutation. But I told them, this is just biology, genetics. It's not their fault. So that I want to convey this message to you also. And how does this occur? This is because of DNA replication. So DNA is a genetic material that need to be copied. And then during, cell division. So during this copy process, there is a mistake. Let's say when we copy something from a paper or from a text, we make mistakes. The cell make mistakes also. So that occurs randomly. So this is a random process. So I just want to emphasize this. And then taking home message, I think the previous speaker also mentioned. So instead of KAT6A foundation, one potential is just to say KAT6 foundation, because you have KAT6A and KAT6B. Their functions are very similar. Not only that, they are part of cellular complexes. I will tell you later, there's another protein. It's very important to regulate the function of these two proteins. So we can include those families affected by KAT6A, KAT6B, BRPF1, maybe potentially even the other proteins like NG5 and NG4. So in the cells, KAT6A itself, the function is regulated by other proteins. So they are molecular machines. So for example, here you have the KAT6A, I also call the moth, KAT6B moth. So basically you have this BRPF protein, previous speaker mentioned, activating KAT6A or B to modify the epigenetic material. So then, so this basically is like modify the, how should I say, the histone. So for that, I think we have the DNA sequence, right? So we have this long DNA sequence. So that forms a genetic language. It's just like me, when I came to the US to learn English, I found it's very challenging. So English is a language. So DNA, that form another type of language. It's genetic language. So now you have the epigenetics, basically modify the histone proteins. Because the DNA, I'll show you later. So you have the English is like a language. So the genetic material also forms this called epigenetic language. So you have the genetic code. So then for the epigenetics, there's another type of language. Of course, that's more complicated. So that's what we call epigenetic language. Basically, KAT6A is just like some kind of writer to write this kind of language. And then the protein formation, BRPF1, is basically activator of KAT6A. So then we have the biology and the environment. So biology is genetic determinants. So then the environment, that's basically linked to the epigenetics. So in the human genome, there are about 20 or 40,000 genes. About 600 of them encodes epigenetic regulators. So that corresponding 2 to 3%. And so for the epigenetic, I think you're familiar with the chromosome. So each cell has 46 chromosomes. So then you have the DNA there. So this packing process is very complicated. The cell needs the epigenetic language to regulate this process. And then for that, the basic unit is something called the nucleosome. So within the nucleosome, you have these histones inside there, 4 types of histone, H2A, H2B, and H3, and H4. So I will focus on H3. So they form octamer, so then DNA wrap around this. So this forms nucleosome. And for KAT6A, actually, it's very interesting. Our research indicates that so this KAT6A mainly modify one particular amino acids of histone H3 at lysine 23. So this basically it's kind of it's like a code, So you have a genetic code now, although it's not exactly another type of code, epigenetic code. So basically, you have the, let me see. So 23, so that then the code can be read by other cellular machinery. So for this, for the acetylation, so if you look at the acetylation, basically you have the amino acid add this ester group. So this ester group, actually we all drink beer and wine. Its chemical structure is very similar to alcohol and vinegar. They share this structure. So this just gives you an idea, for you, some of you never learned biology, so this gives you some idea. So it's relatively simple. You have this group added there. So then for this acetylation in the human genome, actually there are only 14 or 15 or 16 players. So KAT6A and 6B are 2 of them. You can see this is a very important part of this epigenetic language. So the first protein, KAT6A, actually was identified by a group in Boston. This was many years ago. And the gene initially was identified in a patient with acute myeloid leukemia. So in that patient with this chromosome translocation, And one of the genes rearranged is this KAT6A. At that time, it's called MOS. And we identify another protein called MOF, and that is very similar to MOS. So now it's known as KAT6A. So from here, you can see the structure is very similar. So come back to this early point. So KAT6A and B are part of this molecular machine. Okay? And this protein, BRPF, activates this. So it's like me, sometimes I don't want to work. I need somebody to tell me, it's like activator. So things like that. So this basically, in the cell, the protein doesn't work by itself, need other things. It's like in a society, we work as a group. This is the KAT6A foundation. It's like a group, right? It's similar. So then what's the function of BRPF1 in vivo? So a former student who used mice as a model to figure out what's the function of this protein. So she is now a professor in Shanghai, a well-known university in Shanghai. And she made a mutant mice, and the mutant mice contain only one allele. We didn't see any major phenotype, but both alleles lead to lethality at the middle phase of the embryonic development. As you can see here, the blood vessel disappeared in the mutant. And one major defect is the brain. So the head part mainly disappeared. So that was interesting. So she generally a special mice only get this gene deleted in the forebrain of the mice. From here you can see that the protein is highly expressed in certain region of the forebrain, especially in the hippocampus. I'm not sure you're familiar with that area. So that's the area very important for learning and memory. And so then those mutant mice, the lost one allele didn't cause major problems, but lost both alleles caused this lethality like early, just first few weeks after birth. And from here you can see they also show this wild type and this mutant also show some neurological defects. And very luckily, through genomic sequencing and also collaboration with different clinical labs in the US, in Canada, and also in Europe. And we found over 10 patients contain mutation only in one copy of the gene. Of course, from the mouse study, we know that if both alleles contain mutation, so those will cause lethality problem. So those patients, those kids have some, I think phenotype was not as severe as kids with KAT6A or KAT6B mutations, but they do have problems. Share some of the clinical features, especially like intellectual development. I should emphasize here that we call the intellectual disability. This is mainly based on certain indexes. I'm not sure that's the best way, because in intellectual, it's very broad. I also wonder, some of the kids may even have a special talent. So that's a question I want to ask you guys, check your kids if they have certain special ability. Because like me, for example, I'm good at learning at school, maybe mathematics or physics or chemistry, but I'm not so good at music, for example. I used to hate certain things. So for that, like music ability, I'm intellectually disabled, right? So I wouldn't say, use the intellectual disability too broadly. just to define these kids. So I would also, think, if you check the history, certain of the painters, musicians, they have special talents, but they lack certain aspects. So this may be also an encouragement for the kids with the mutations. So that may be perhaps the best message I can give you today. So then for the molecular models, we think that loss of this one allele causes this histone modification, the check affect the epigenetic language, so then affect the development. And so the next question I had was whether there are any treatment options. There's one potential drug called the valproic acid. This was an old drug used by clinicians since the 1950s for epilepsy. So this is a drug because those kids, mutation usually decrease the escalation. So this can boost the escalation. So then there's another compound called sodium butyrate. So this is a very interesting compound. If you look at the chemical structure for the ester group, this is just a little longer. So this compound actually says a lot in our statin. And from bacterial fermentation, especially when we take non-digestive fibers. So this actually is also important for health. So that's one thing. And for the last, I think also, while I came here yesterday, I was thinking, I just considered this thought experiment. What if I had this mutation? So what I would do? So the question is that if I was one of the parents of such a kid, so what I would do? So I think check, if the kids have any special abilities, and try to focus on developing those. So related to that, I studied brain development. Brain is a special organ, has something called plasticity. So for example, for my finger, genetically determined how long, this is very determined. For the brain, it's flexible. For example, I can learn English, learn math, so it's flexible. So it can change a lot. So there are a lot of potential. So that may be something parents can think about. Okay, I will stop here if any questions. Thank you. Yes. Yes. So currently, we from the cells, actually, I didn't mention that we also found those KAT6A actually has a new function. So we are using those drugs to test those also. So yeah. So for patients, I think we need to organize a clinical study. For mice, it's easy, yes. Patient, I think that's something I'm considering because I have a collaborator. I forgot to mention. Yeah, for the collaborators, Dr. Kapoor is the clinician we collaborate and many other clinicians. And also, I should mention that here, for that study, there's another study, independent study from France, and reported similar results. So yeah, I think that's one possibility. I think so, because that's already a drug. Yeah.
We have some children here, actually, family from Canada, and we know a few families from Canada who are much interested.
I think that's one possibility. I can discuss. Yeah. Yes. Yeah. Yes, yes, yes. Now, I think this thing, for example, I think different individuals have different abilities. We just need to somehow find, because for me, I cannot do everything, right? I do the things I can do, I'm good at it, right? That's true, yeah, that's good point, yeah. Okay, thank you.